Then I'll tell you about a model which we have developed, which explains why the immune system we think is scale invariant. I'll point some trade-offs between local dynamics and global dynamics, and this is going to be a persistent theme throughout my talk. I mean, these trade-offs will also present themselves not only within the immune system, but also in other human engineered systems. And I'll use a simple time minimization argument to show how you get an optimal arrangement, and then I'll apply it to engineered systems. Okay, so here's again a very cartoonish depiction. Again, I see some new faces, so I'll just give you a brief overview of what's going on. So there is an arms race. This again is a very simplified view of the immune system, but there is an arms race between the host immune system and the pathogen. So here is my virus here. It's injected in this case by mosquitoes. Then it infects a normal cell, which becomes infected, produces more virus particles, which then go on to infect more cells. And this feedback loop ensures that there's an exponential replication of virus initially. And initially, the only thing stopping this pathogen from killing everything is this first arm of the immune system, represented by this macrophage cell. So this cell literally recognizes these virus particles and gobbles them up. There is a certain delayed response which comes in later. There are tailor-made weapons made by the immune system against the virus, which secrete these chemicals known as antibody. Again, this is going to be a critical part of our modeling effort. But the picture that I want all of you to have is there is an arms race between the host immune system, which is always trying to play catch-up with the pathogen. And we expect from biology, we expect things to get slowed down in larger animals. So metabolic rates are slower in larger animals. So if you look at the cell in a mouse, it has a higher metabolic rate than the cell inside the elephant. And that has implications for how fast this virus which infects the cell is replicating itself, how fast it's turning out virus particles from an infected cell. That also can affect the immune system. We expect immune system cells to be slower in larger animals. Apart from that, there's a problem of, so if you look at mice and in elephants, in each of these, there are only very few immune system cells which can recognize a specific pathogen. So if you have like a specific strain of influenza virus, and I'm simplifying it again very vastly, but if you think of any virus in the world as a lock, the immune system has a key which unlocks it. And naturally, I mean, if you look at the whole universe of pathogens, there are millions of them. So that particular key for that particular virus is very rare. So there are almost 1 in a million of those specific to a particular virus. So the search problem is harder in elephants because those immune system cells have to search throughout the physical space of the body of the elephant, and that physical space is much smaller in smaller animals. And then the immune system has to respond by producing antibodies. And the problem with antibodies is they get diluted by blood. And larger animals have more blood, so there's more dilution, which means that larger animals have, not only do they have a hard search problem, they also have a hard response problem. They have to produce more absolute quantities of antibody also. And last week I showed, how using machine learning techniques and other techniques that we have developed, we showed that fitting very sparse time series data to infection data from different birds, we showed that the rate at which infected cells release virus particles is actually slower in larger animals, which is precisely what we predicted. So since larger animals, so here is mass logged axes here, and then that rate again logged on the x-axis. And you can see larger animals are producing virus particles at a slower rate. That's what we predicted, and we expect that because of various theories from biology. What we don't expect is this. This is the time taken by the immune system to actually respond, and this is also true for rates. So the time taken by the immune system to actually mount a response by secreting antibodies or find specific pathogens does not scale with your body size. Suppose here's an elephant here or a horse right here that is able to find and respond to that particular pathogen in the same amount of time as very small animals also. Naturally, as a computer scientist, this was something very, very intriguing. And today what I'm going to tell you in the first part of this talk is, how does the immune system achieve this? And I'll tell you that there are physical architectures which facilitate that. And can we use that as an inspiration to build something nice? And so how does the immune system achieve that? And the answer lies in these small anatomical structures called lymph nodes. And these are, you would normally find them under your neck, under your armpit, and they form a decentralized network throughout your body. And they are like distributed detectors. They are more or less evenly distributed throughout the body. In some places you can see that near your gut, for example, there are more of these, especially because the gut is taking in a lot of food. There's possibly a lot of foreign bacteria and pathogens which are going into this. So there is larger concentration of lymph nodes there. But then, outside it, it's more or less evenly distributed. And this is what makes search in the immune system. It makes it modular, so it's absolutely distributed, and it's also parallel. And what I'm going to show you here is that you need a specific architecture. So here's my hypothesis, that the architecture, there's something interesting about the architecture, how these are organized, what numbers they are found in, what their size is. There's something interesting about that, which is responsible for what I call scale invariant search and response. And I should emphasize that it's not only the architecture, it's how these immune system cells also cooperate with each other, how they signal amongst each other, and the strategies that they use to actually literally hunt for virus. And again, since I should mention that this is just for West Nile virus. This is a virus which initially infects a small region of tissue and then spreads. This picture could be very different for something like dengue or malaria or bacterial infections. We don't know how the immune system precisely responds, at what time points do virus show up in different places. It's quite unknown. And so here's a model of the lymph nodes. So lymph nodes, there's a joke in our community. It's called circular cows. So it's easy to model cows as little circles or spheres instead of looking at the actual shape of that. So sort of in that vein. So I'm modeling lymph nodes as like a three-dimensional spherical object. And there's a region outside it. There's an area, or there's a volume of tissue which is literally patrolled by this lymph node. That's called its draining region. So just a particular lymph node is literally in charge of that region, and what it does is, there are cells in that region. So in this case, there is this dendritic cell. It has these dendrites which it's constantly pushing out and literally probing for foreign things. And when it finds something interesting, it says, aha, I've found something. Let me just travel to the nearest lymph node. It literally carries this, I'm obviously simplifying, but it literally carries this virus on its head. And it says, you know what, I have found something very interesting. I cannot do anything about it, but I think this is something that you should look at. And so it goes to its nearest lymph node. And this lymph node is actually secreting chemicals which lets immune system cells know where it is. So it can almost make like a straight beeline for its nearest lymph node. When it does that, it actually has to, what it's called, present this virus to immune system cells. And here is that 1 in a million, here's where that 1 in a million thing comes in. So here's an immune system cell, it's called a T cell, which is actually specific for the virus which the dendritic cell has captured. And the thing is that this T cell is very rare. This is where that 1 in a million thing comes in. It is present in the body somewhere, but it's just present in the frequency of 1 in a million. So getting that particular T cell into this lymph node, and then this T cell has to search for the dendritic cell and finding it is tricky. And once that happens, then what needs to happen is that this lymph node literally tells other lymph nodes to, it starts recruiting other immune system cells towards it. And it says that I need to build up my numbers because you have only found one immune system cell which can take care of one virus. But naturally you need to build up numbers. What happens after this is that this T cell then starts building copies of itself. It goes through a process, what's called clonal amplification. It clonally multiplies, but you need more of them to actually show up, and that process of multiplying itself, that takes time. So here is a very complicated search process right there in the draining region. Then there's a migration process, then there is a detection process within the lymph node, then there is recruitment, then there is replication to build up enough numbers, then it has to secrete antibodies. So it's a multi-step process, and our work suggests that this happens in more or less the same time in smaller and larger animals. So more or less within 2 to 4 days after infection. So if you get like a flu, if you get any kind of infection, then within 2 to 4 days your body is able to mount an immune response. And think about the implications for what would happen if mice were able to detect and respond to influenza in 2 days, but you took 2 weeks. I mean, you wouldn't be there to even just survive that. So there are very strong selective pressures at work to actually make this very, very efficient. And that is one assumption which I think is justified, that the immune system is under tremendous selective pressure to be very, very efficient at what it does.
When you say efficient, you really mean fast.
Yes.
It may consume huge resources.
Oh yes, absolutely. Absolutely. I mean, that is, you make a good point. I mean, so the immune system does consume a huge amount of energy. So from an energetic viewpoint, it may not be, efficient, but that's a good point. It consumes huge amounts of energy, especially during infections. And then there's the process of actually maintaining that immune system even when you don't have an infection. So yes, a good point though. Yes. So again, I just want to repeat this point, but this is crucial. Why do lymph nodes have to recruit immune systems? So if there is a single cell which has found its virus, it can't do the job of actually eliminating that. It has to recruit, because in say a horse, so a horse is 25,000 times larger than a mouse, which means that if a mouse has say around 3 milliliters of blood, a horse has 75 liters, 70 liters of blood. So any antibody that you secrete in the blood of a horse is getting diluted that many times more. So you need to secrete 25,000 times more antibody in the same time in a horse. And so, again, this sort of assumption is that you need a fixed antibody concentration in blood to actually neutralize virus, and that's why you need to produce more antibodies. And so what needs to happen here is, so suppose you have some fixed amount of lymphatic tissue, some material out of which you want to make lymph nodes. So given some material, you can divide that up and make a few larger lymph nodes, which means that so these larger lymph nodes are now in charge of like a larger draining region. So they have, so if this is a lymph node, they are in charge of a larger region now, which means that any dendritic cell which finds something in its region has to travel over a larger distance. So you're literally far away from, suppose you get an infection in your knee or on your foot, and there's a lymph node like in your arm. So that dendritic cell has to travel like 1 or 2 meters to actually get there. Naturally, that's not desirable. But larger lymph nodes also have an advantage. They have what are called high endothelial venules. So they have literally more. They're literally more porous. So they can recruit more immune system cells. So if you have larger lymph nodes, you lose out in the local detection process. But what you gain is you are able to recruit more immune system cells. So if you have, now say, you want to divide that same material up into lots of small lymph nodes. So now what happens is, so each lymph node is smaller, there are more of them now, and so each lymph node is in charge of a smaller draining region. So that migration time is now shorter, but each lymph node doesn't have enough vessels to actually recruit more immune system cells. So it wins in local communication but loses out in global communication. So one theme throughout this talk is going to be this trade-off between local and global communication. And there are, that will ultimately dictate what kind of size is actually optimal, what kind of numbers that you. I just want to pause here for a moment to see if that makes sense to everybody. Okay. So there is this trade-off, and so what we did was, this is this multi-step process that I described. The time taken by the immune system to actually detect and respond is the sum of the times for this cell to detect, then to migrate, then for this cell to actually make contact with its specific pathogen, and then for this lymph node to actually recruit more immune system cells. And so what we did was, we set up an agent-based model. So last week I talked about these differential equation models, and those assume that everything is well mixed. So you just give a box, you take a box full of two things, give it a good shake, everything is well mixed. Unfortunately, within the lymph node, even outside it, things are not that simple. So this is data that we took from actual experiments. So here is a screenshot of what this agent-based model does, and it's really crowded because that's how many. So these red things, or these red agents, are actually immune system cells which are in that draining region, and it's really crowded because there are a lot of them outside. And then this green, or whatever color this is actually virus. And then once these immune system cells find it, they're actually moving towards a lymph node, which you can barely see in the background here, in that bluish hue. And so we model each virus, each cell, and the whole lymph node as separate agents. We are not looking at populations now, we're looking at individuals. And then we simulated different sizes of lymph nodes and looked at what those times were and whatnot. And what turns out is, here is it's a very simple optimization argument here. It's so simple that I'm sure I can explain it within a few seconds. So the time here is now roughly proportional to the volume of the lymph node here, raised to the power of 1/3, which means that is actually the migration time. And the migration time is now proportional to the third power to the volume of the lymph node raised to a power of 1/3, which means it's proportional to the radius of that lymph node, or that draining region. So that makes sense, because if this dendritic cell, which has just found something, has to migrate towards a lymph node, it takes a straight line towards that. You're going to travel as a radius, and the radius scales. These are the radius scales. And the time taken to recruit is now proportional to the, inversely proportional to the square of the lymph node volume, and that immediately sets up this trade-off which I mentioned. And so, doing a very simple optimization, we found that the number of lymph nodes should scale sublinearly with the size of the animal. So capital M here is the body mass of the animal. It should scale sublinearly with body size, and also the volume of the lymph node should scale sublinearly with body size. What do I mean by sublinearly? So let's say a mouse has 20 lymph nodes, and a human is, say, 100 times larger than a mouse. It should have 2,000 lymph nodes, right? That's what linearly predicts, but it has much fewer than what you would predict from just a linear. So we have some data. So yes,
this minimization, you still have to conserve something, right? Because if you could populate the whole body with lymph nodes, then the time essentially is 0.
Yeah.
So what is the resource? What is the resource?
The resource is lymphatic tissue.
Okay.
So
you still try to keep it as low as possible, right?
I try to keep it linearly as makes sense.
So that's another constraint.
That is one constraint, and that we do is because that tissue is actually energetically expensive. So in order to maintain that tissue, there are some data showing that the immune system is actually investing energy linearly with body size. So it's investing more energy sort of in larger animals and linearly more in larger animals. So the assumption is that lymphatic tissue is actually conserved, it's energetically expensive, and that's an assumption which.
So to some extent, the immune system actually is a little bit of an agglomeration economy, right? In that,
Yes.
because of the size, you can actually afford to have less lymph nodes.
Yes.
That,
A little less, yes. A little less, but make them bigger.
Interesting.
And so what happens is, so we have some data showing that, so suppose you have a mouse, and the mouse has a really small lymph node, like 1 millimeter cubed. Elephants have lymph nodes the size of mice. Literally, I mean, its lymph node is the size of a mouse, because it's much bigger, but they're not the size of like horses. I mean, they're not that big, but they are, I mean, how big is it? I mean, that's dictated by this exponent right here. So we make predictions for how big lymph node sizes should be, how many numbers should they be, and they are roughly consistent with some data that we have. But I should caution that, I mean, we definitely need more data on larger animals. So what does the picture look like? So suppose this is a mouse, and this is a human being. Let's just play that spherical cow or cubical cow game again. So this mouse has 4 lymph nodes, and here's the time taken or the path taken by dendritic cell to actually migrate to that lymph node. Here's the draining region of each lymph node shown. And this lymph node, suppose that has found something, then it starts recruiting immune system cells from other lymph nodes. And here is the amount, so this, so the length of this arrow or the width of this arrow shows you how much recruitment power, how many immune system cells it can recruit. And suppose that this is a human being with now 9 lymph nodes, and which are much bigger than those, each of which is much bigger than that lymph node. Yes?
And if the question is too complicated, skip it. So let's say that you are bitten in 2 different places. So 2 different lymph nodes detect this at the same time, and they are all sending each other a request for collaboration. Does it ever happen?
That happens. Great question. I mean, I have like a list of 10 other research questions that I want to investigate. That's a great question. It's really unknown, and I've talked with immunologists. It's really unknown what happens when that particular scenario happens. I mean, what you mentioned is, I mean, could be going on. I mean, suppose you get 2 mosquito bites,
right,
in 2 different places. What happens?
Roughly at the
how
same,
do they communicate?
Roughly at the same time, right?
Roughly at the same time. I mean, how do these lymph nodes actually communicate? I mean, is lymph node trying to dominate one another? I mean, what happens if one lymph node gets sort of priority over, over the others? I mean, this is not only a nice biological question to ask. I mean, as you can imagine, I mean, there are implications for how you design systems in which there is some kind of, uncertain event. I mean, how do these different components, which are, are actually built to maximize or minimize time to response, how do they actually coordinate with each other? It's really unknown. So for something like dengue, which spreads by blood, it's probably safe to assume that it infects most lymph nodes in the body simultaneously. But it's not known, I mean, what happens in each lymph node. Are they recruiting simultaneously, or does one lymph node say that, I'm in charge, let me handle this? But that's a good question. I mean, one which we don't know the answer to, but we should. So here's say a bigger animal with 9 lymph nodes, each of which is now larger than the lymph nodes in that mouse which I showed. And now, since this is this larger, it has a larger draining region. This dendritic cell has to migrate a larger distance, but then this lymph node also has a larger recruitment power. So again, this width of this arrow is larger than the width of that. Or it should be, at least. I don't know if it shows up from here. But so this lymph node is faced with the challenge of having a larger migration time in its draining region and a larger recruitment. But if you had like a single lymph node as big as this in the middle, it would have a huge recruitment power because everything is just within it. But then all these dendritic cells have to travel from distant corners. So that's a trade-off there. And I'm not going to be able to talk much about this, but it's not only about the physical architectures. So I told you how many lymph nodes, how big are they, how are they organized, and that plays a role that reduces times to response. And we showed that that alone can lead to scale invariant response, just the plots that I showed you initially. But then there are the signals also which play a role. So we looked at, and this has been almost a 3-year project working with multiple experimentalists on strains of influenza now. So there's a different virus. So this is influenza within the lung. And again, this is sort of different because influenza is only in the lung. You see some antibody in blood, but antibody almost, it's safe to say that it doesn't play as higher role as in Western. So different virus with different dynamics. And what's going on here is, so influenza in human beings initially produces this tiny zone of infection which is 1 millimeter squared in area. But the size of the human lung, if you spread it out, if you can spread it out, is the size of an entire tennis court. So our research question initially was, okay, suppose you have lymph nodes which are strategically positioned and they have this nice structure. This immune system still needs to do something very different for it to find that tiny zone in that tennis court region. And our first clue was when we started working with virologists who study this, that this tiny region actually sends out signals. So it's literally like a beacon, but it's in a tiny zone nevertheless, but it's still sending out signals that, there's something funny here, I need help, please send me immune system cells here. And so the schematic, which is what's going on sort of within your lung once you get an infection, is suppose there's this lymph node which first senses that there is an infection, it's sending out immune system cells which then have to traverse this sort of hierarchical tree-like structure of your cardiovascular network. So your blood, there's an aorta which comes out of your heart that gets divided into multiple pipes, each of which at successive levels are getting smaller and smaller in diameter. And ultimately at this very end of this tree, you get down to tissue. So I said that this region is sending out signals, but it's a local signal. If you're an immune system cell somewhere up high in this tree, you actually cannot sense where the signal is. So it's sort of a signal, but it doesn't give you that much help. So we did a lot of modeling coupled with detailed experimental data. I really don't have much time to go into this, but we looked at agent-based models and differential equation models, and we predicted that these signals also speed up search and response within the immune system. So there's a physical architecture of the immune system, and then there are the signaling strategies. Since I'm a computer scientist, one view that I want to impress you with is that we view the immune system as a computational system. And if you buy that, which I'll try to convince you, then the structure of how the lymph nodes are organized is the hardware. And how these immune system cells actually communicate with each other is the software. So we talk about hardware-software evolution, and that has been going on within the immune system for millions of years. This hardware, how these lymph nodes are organized, that has co-evolved with how these immune system cells actually communicate with each other. And this is just me summarizing this part of the talk. There are increasing costs to global communication as these animals grow larger. And you find these trade-offs between local and global communication, which leads to this very particular structure. And the question is that, can we emulate this strategy in distributed systems? Yes, we can. And here I'm going to tell you about two different systems. But before I say, we looked at actual systems which are implemented, but we make theoretical predictions. So we did not actually build these systems, but we looked at systems which were actually implemented, and we said, how can we modify this to actually get gains? So the first thing that I want to talk to you about is a simple peer-to-peer system. So peer-to-peer systems, for those of you who are unacquainted, these are systems which are massively distributed. computer nodes which store some data or service. So the search here is not for a pathogen, but the search here is for presumably rare amounts of information. And no single node, so let me go to the next slide here so that I can show you the system. So here's a cluster of computers, and these nodes are single computers which are physically close to each other. And so these nodes here form a cluster of computers, each of which, so within a cluster there are very, very tight connections, and then distinct clusters are separated by distances. And this particular system that we looked at said that you can speed up search for rare amounts of information by ensuring that connections between two different clusters follow what's called a small world distribution. And this follows John Kleinberg's very famous results from theoretical computer science, saying that if you are doing what's called decentralized search, so if different nodes have information, but they can, so if I'm a node, I can communicate to my nearest neighbors, but I don't have global knowledge, then you want this kind of a distribution. You want to draw your degrees from a small world distribution. And the reason that you don't want to have global information on each node is because that's a lot of information. I mean, you're talking about millions of computers storing gigabytes or terabytes or petabytes of data. You cannot store, it's very, very expensive to store global data. So you only have local knowledge, and then you're what's called local routing or decentralized search. So that's what this is this very well studied system, and it's implemented almost everywhere now. There's a system that we are looking at. And what we said was, there is a way that you can modify it to actually speed up search. So what's happening is, there is a global communication cost here. And by global communication cost, I mean the time taken to actually route one message, or the time taken to actually find a message between clusters. That's what I mean by global. And this is a result which I pulled from this, just from the people who actually implemented this. So their result was that the global communication cost should scale as O of log squared n divided by n. So n here is the total number of nodes. So as the total number of nodes in the system increases, you get increasing global communication costs. That makes sense, but what if you divide those numbers up into clusters? So if you divide these numbers up into clusters, so you literally now have fewer nodes to communicate with. So this cluster does not care, how many nodes it's communicating within this cluster, it just cares how many clusters there are. So the idea is, as you make the system bigger, if you increase the number of clusters, you can balance that cost. So again, balancing local costs within the cluster, there is what's called a flooding process. You just send out messages to everyone. I want this information, everyone within my cluster, get me that. So it's a very simple cost. But between clusters, there is this global communication cost, which scales as a number of clusters. So the idea again is to make these clusters, these lymph nodes, larger in larger systems. Again, this trade-off between a local and a global communication cost. And what turns out is you get and we did that same minimization. Let me show you what that minimization was. So the time taken to actually find a single piece of information is the sum of the local and global communication time. The local scales as the square root of the size of the cluster because it's flooding. And the diagonal, if you place all those clusters in a grid, all those nodes in a grid, the diagonal is the time taken, minimum time taken to actually find that. So that's how local communication cost scales. The global is a function of n over c. Suppose you have n computers, 100 computers, and you have 10 clusters. What you do is you're literally now communicating with 100 over 10. You're communicating with 10 clusters, not 100. So this is the reduction in global communication cost. You increase the cluster size here, you gain a little bit there, but you can actually gain a lot here also. So there is this trade-off, this tension again between local and global communication costs. And what turns up is a system very similar. So if I look at it from the lens of using the immune system perspective, what happens is, so here's a smaller system, one cluster with 3 computer nodes. Here's a larger system with more clusters, And these clusters, each of these clusters is now bigger. So it's like a bigger lymph node. Each cluster has more computers within it. That's how we predict this system should scale, and we actually see a speed up in that search time if you use this architecture.
So why are you only considering one level of aggregation? I mean, why isn't it recursive the way you're saying?
It should be. I mean, that's my future work. So make it fractal.
Yeah.
Absolutely. Make it, what is this?
But
This is a cluster.
biologically, does that make sense as well? I mean, is that
You know,
how it's
that's a
being
very, again, I mean, that's a big sort of known unknown. I mean, we don't know if that makes sense or not. I mean, I think it should, and lymph nodes are actually sort of organized hierarchically, but is one lymph node in charge of the liver, and within each liver is like smaller lymph nodes?
Yeah.
I have no idea. But you're right, I mean, recursively, I mean, from a computer science perspective, I can make each of these nodes like a cluster, and then within that I can recursively keep on applying this, and you will get even a larger speed up with presumably some cost now with maintenance, because now you have to maintain those links.
And then there's no experimental data about how the lymph nodes communicate. Is there?
There's
No whatsoever, because then you could infer, try to test these models, right?
Yes, but there's no experimental data.
It's very difficult.
It is very, very difficult.
Because why is it? Because they don't have so the signals don't have like an original destination.
They don't have an original destination. You can image the immune system, so you can look at a live organism, but you can only look at lymph nodes which are near the surface. If you want to look at like lymph nodes within the liver or something, you literally have to pull the liver out, and once you pull that thing out, it's no longer the same thing.
Yeah.
But if we could find that out, I mean, that could, I would argue, I mean, that could form valuable inspiration for designing systems like this. How do lymph nodes actually communicate?
Well,
There.
I mean, if you can do that, you could win a Nobel Prize by testing
by testing.
by testing how it works. It's not only inspiring.
Let's see how I'm doing on time.
Let's say 5-10 more minutes.
10 more minutes?
Yeah.
Okay. Then let me just skip ahead with this one. We also looked at multi-robot control. So these are robots in which there are computers which are in charge of a single region. That region is populated with robots. You can see where I'm going with this. This computer is like a lymph node. It's in charge of computers. How should they communicate? We think there should be trade-offs between local and global communication costs. That's a theoretical argument, so I'm working with some people here. This is what's called an antibot, or it's got an iPhone on it. It's got GPS and all other fancy stuff on it. This is one of so this robot was made by one of my collaborators in my department, and so I'm working with him to figure out if we can test these predictions in a real implemented system. So working on that.
What's the advantage of having a physical implementation as opposed to just a simulated one?
You know, that's so. I mean, I think that, so again, these are theoretical predictions, but what could be going on is, one assumption is that these regions have to communicate with each other. Does that even make sense? I mean, here I'm making a theoretical argument, but what sort of advantages would robots get by communicating with each other? One scenario could be that these are very different regions. This could be like a hilly terrain. This could be like similar to a hilly terrain, or some other. There could be something in there could be some local information which is valuable globally. And I think there's merit in actually seeing if that even makes sense from like an engineering perspective. It's easy to sort of say that, okay, we can do that, but does it even make sense? In what scenarios is that even applicable? So that's a like an engineering challenge to see if we can really put this to work, and if somebody can make use of it. Okay, then we are doing more work again with the same person, looking at how you do distributed load balancing, again in sort of distributed systems. You have a distributed an operating system, and these are, you can think of computers one particular computer that's handling requests for CPU time to different processes spread out throughout the system. And again, you can get the lymph node idea. I mean, one of these could be controllers and controlling how those requests are handled. In this case, we're interested in seeing if the time to allocate CPU to processes as the system size increases, can you keep that more or less constant, or 1. Then there are applications to intrusion detection systems. You can see there is a ton of systems that we can look at, each of which has different constraints. I mean, it's not necessary that in each of them you will find this kind of trade-off between local and global communication. So I have here as these kinds of system requirements. What kind of system are you looking at? What is objective? And then you look at it from the lens of the immune system, and magically your problems disappear. I'm joking, but I think there are some it's not as simple as that. But I think there are merits to actually viewing distributed systems from the lens of the immune system. Since it has an architecture, it has a strategy, there are hardware and software decisions which can be informed by them. And so one open question which I want to talk to you about today is, how do immune system cells actually replicate in response to immunity? So I told you that there's this one immune system cell you start out with, and then it starts building copies of itself. But making copies is energetically expensive, and then these immune system cells actually secrete chemicals. They actually attack normal cells also. So they can cause a lot of damage if this process goes out of hand. And indeed it does. In influenza, which kills the young and the elderly, this is what happens. So the influenza virus is doing damage in the lung, but the immune system cells mount a disproportionate response to the virus, secretes chemicals which actually block up your airways and destroy your lung, and that's ultimately which kills you. That's what kills you in pandemic strains. It's literally not the virus itself, it's this hyper-reactive response to the virus. In some cases though, your immune system mounts a proportionate response. It matches the punishment to the crime. But how does it do that? It's really not known. So how does the immune system know when to stop? It's really not known. So how is the magnitude coupled to this response? It's really not known, and this could enhance our understanding of systems similar to the immune system. So over the last 2 or 3 weeks, how time flies, I've been working with Manuel on looking at crime as a competing system, so criminals and police competing with each other and their dynamics, ultimately explaining crime in cities. And so one thing that we have been thinking about is what happened in the London riots a few years back, or just a couple of years back. So the police knew that there was all this rioting happening, but they probably could not respond quickly. They could not ramp up their numbers in time, which was critical to actually suppress that response. So with Manuel, I'm also interested in questions like, can you design systems where the time of response scales very efficiently? So here you have the size of the system increasing, the time or the rate at which it's responding is invariant with body size. Can you design human engineered systems which do that? And one thing that we are looking at is, how does police response scale? Some preliminary results that we have been getting says that time of police response probably increases in larger cities. So if you look at larger cities, the time taken by police probably increases with the size of that city, and so there could be more crime, indeed more crime, and we see that there is disproportionately more crime in larger cities, possibly due to this instrument. So this is also has implications for how you design systems. Again, my sales pitch, I've told you all those things before, but just want to say that all these results and this work was recognized by an award for innovation in informatics from my university. 5 papers published, 3 more in review in top journals in biological and computational biology journals, and you see I have a few other projects in the pipeline which will give more results hopefully. And just want to thank all of you and Manuel and Pascal for inviting me here. Thanks a lot for listening so patiently. I've had a great time here. I hope to visit you again soon. Thanks a lot.
Thank you. Time for questions? have one.
Yeah.
If you can go back to when you optimize and derive the scaling relationships. Yeah. So you make very specific predictions here about the number and the volume of the lymph nodes, but you don't. Do you make any predictions about the positioning of them?
No.
Because that's important, right?
That is
We're talking
very
about the
important.
physical system.
Yes.
Because even Kleinberg says that it has to scale with the square of the distance, right?
Yes.
So do you have a conjecture or where they should be in the body?
We thought about, I mean, when
That's too complicated, maybe.
Yeah, that is complicated. But yes, I mean, one sort of research question that we had is that what kind of connections are there between Kleinberg's decentralized search model and how search takes place between different lymph nodes? Is there like a small world network? Is that how lymph nodes are connected? But then, I mean, one complicating factor is even though lymph nodes are organized in this hierarchy and there is this, I think there is valuable information in space, all of these lymph nodes are literally bathed in blood. So blood is like, that's what made us think that probably that small world model is not right. But that's one complicated, because that's like a shortcut for all the lymph nodes. All of these lymph nodes can literally dump anything in blood or recruit directly from blood. So there are some complications there. But I think there is this valuable information in space. It's possible that there
Because
are...
once you have less, you need to know, if you have a lot,
Yeah,
it doesn't matter where you put them.
that's a good
But
point.
once, because it scales sublinearly, once you have less and they're bigger, it does matter,
That's a very good point.
right?
So you're saying that in larger animals, I mean, larger animals could be exploiting the spatial nature of how these lymph nodes are organized. That's
I mean, I have no clue, right?
No, but yeah, that's a
Yeah.
great suggestion. Yeah. So I spent months looking at veterinary journals, looking at how lymph nodes are organized in cattle, in sheep, in dogs, and other things. And it's very complicated because some of your lymph nodes are actually larger than others. So there's a lymph node right here,
Yep.
which is the largest lymph node, and those lymph nodes are organized differently in cattle. So I spent a lot of time looking at these arcane journals, which I don't think people would.
But the variability comes from intra-species or across?
Both. So even within a species, so you could look at like sizes of, say, a calf versus a cow or something like that. They have different organizations. Probably I don't know if you get lymph nodes later on in life, but probably within puberty you do see more lymph nodes coming up, and then you sort of hit like a threshold level beyond which.
So nothing is known about the distribution of the distance between them? Interesting. Any more questions? Well, thank you again.
Yeah, thanks a lot.
If we have some more, we'll take them offline. Thank you
Okay.
again.